Thank you very much, Radek, for your kind introduction. Good afternoon, ladies and gentlemen. I've got half an hour to get through a lot of material relating to essentially longer-acting products is going to be what I'm going to be talking about. As I said yesterday, you're very welcome to take copies of my slides from the computer at the back. Alternatively, if you write to me by email, I'm happy to email you a PDF copy of my presentation. Now, the questions I want to answer are here. Firstly, first half of my talk is about the new products themselves, an update on the new products. But really the second part of my talk is on these more interesting questions. I'm not going to read all of them out, but I want to touch on what are the practical implications, how we're going to use these products in the future. So, as I say, the first part of my talk is really an update on the new products. We're going to be hearing a lot about what the potential advantages are from both Claude Négrier and Beatrice Nolan. So I just want to say I agree that these products potentially are going to transform the lives of people with haemophilia. And I think we're going to be talking about a change in paradigm, moving away from trough levels of 1%, although we can discuss exactly what trough level we should be aiming for. Clearly, it's going to improve prophylaxis, so we can start earlier, and it'll probably facilitate compliance, and therefore we'll get better outcomes, and there'll be less need for venipuncture and probably reduced need for venous access devices. I think it's clear to me so far that there's a more obvious advantage with the longer-acting factor 9s than with the factor 8s, because as we'll see with the factor 9 products, we can prolong the half-life roughly 5 times, whereas for factor 8, we're lucky if we can just double the half-life. I think we need also to be aware that although we are very enthusiastic about these new products, there have been several failures and problems which have led to the premature termination of several clinical studies so far. In the past, with conventional recombinant and plasma products, we've always assumed that these are going to be licensed and we just go through the rigmaroles and the practice of doing a clinical trial, but there have been several problems. And I think with the new factor 8 and factor 9s, I need to remind you that nobody has yet started studies on previously untreated patients, the PUP patients. All the data so far are on PTPs, both adults and children, and we don't have any published PUP study data yet, and only that will be able to give us a handle on the immunogenicity of these products. And again, with all clinical products, really the risk of things like allergic reactions and thrombogenicity only become apparent some years after use of these products. But as we'll see, so far, everything is looking very good. So let's just jump straight away into the 3 ways in which you can extend the half-lives of factor 8 and factor 9, and let's look at some of the recent clinical trial data. Pegylation involves covering the factor 8 and factor 9 molecule with polymer chains of polyethylene glycol, and this strategy is being adopted by 3 big companies, Novo Nordisk, Baxter and Bayer, to extend the duration of action of their coagulation factor molecules, basically covering the molecule and protecting it from normal inactivation processes. Now, I don't have time to talk about all the products, but let's just start looking at the factor 9 data because there's some recent stuff that's been published. And in the pegylated factor 9 from Novo Nordisk, the concept is that the polyethylene glycol is applied to very specific parts of the molecule within the activation peptide. And this activation peptide is clipped out during the process of activation, so that removes the polyethylene glycol and you're left with conventional factor 9. And in the initial clinical trial, which both myself and Claude Négrier participated in, and this was published back in 2011, you can see a very extensive prolongation of the half-life, some 5 times longer, to 93 hours, compared with both plasma and recombinant products. Now, the phase 3 data have just been published. This is online in Blood, so I can't give you a proper citation because it's just online. But these are the very latest data which look at the polyethylene glycol from the pegylated factor 9 from Novo Nordisk in 74 previously transfused patients, adults and pediatric patients with factor 9s of less than 2%. Treated either on a prophylactic basis with 10 or 40 units per kilogram once weekly for prophylaxis or on demand. And you can see that in this way, the annualized bleed rate was knocked down as follows. On 10 units per kilogram once a week, down to 2.93. On 40, 1.04. And in the on-demand patients, that was the annualized bleed rate. It's interesting to note that this product was able to reduce very significantly the frequency of bleeds into target joints, more so in the higher dose of 40 units per kilogram compared to 10 units per kilogram. But for me, the most interesting bit is in the next slide, and that's looking at the trough levels. Look at the trough levels. With 40 units per kilogram of pegylated factor IX once a week, the trough level that was maintained was a mean of 27 units per deciliter. That's 27%. And if you have 10 units per kilogram once a week, you get a trough level of 8.5%. Two other interesting things come out in this latest update on the pegylated factor IX. The first is that most of the clinical trial data so far has been using these products prophylactically to prevent bleeds. Here we see clear data showing that it works well in the treatment of bleeds in the on-demand patients, so a success rate of 92.2% for treatment of actual bleeds, which is obviously in line with experience with other standard recombinant products. No inhibitors or safety issues identified in this study, and I think it's great that this company also introduced in the clinical trial an assessment of the quality of life, and we have clear, because this is going to be very important when we come to justify the increased cost of these products, that the quality of life improved using the EQ5D from a median of 75 to 90 with 40 units per kilogram once a week. Now again, forgive me for showing just the Novo Nordisk data, but I emphasize again, Baxter and Bayer are using pegylation for factor VIII as well. Let me say that the pegylated factor VIII study, the phase III study, is in the process of being written up, and hopefully that will appear at the beginning of next year in press. But suffice it to say that the preliminary evidence suggests that the half-life of pegylated factor VIII is, from Novo Nordisk, is about 1.6-fold, so it's obviously far less than the factor IX, but pegylation extends the half-life of factor VIII by about 1.6 with this product. But I emphasize the final the phase III study is being written up, and that should be published, I think, at the beginning of next year. This is just showing similar data. Now, to bring you up to date on the next product, these are products which are linked to immunoglobulin molecules, and these products, as we'll see, are already licensed in some parts of the world, unlike Europe, and I'll come back to that later. So the second way you can extend the half-life of factor VIII or factor IX is to link the factor VIII or factor IX to an immunoglobulin molecule, the antibody molecule. Why do we do that? Well, we exploit the fact that immunoglobulins in the blood have a long half-life, and they have a long half-life because they undergo a process of recirculation and recycling in the body. How does this happen? Well, in this picture up here, what we see is proteins being taken into the endothelial cells lining the blood vessels, and numerous proteins get taken up. Some of the proteins, like immunoglobulins, are able to bind to receptors called the FcRn receptor, and by binding to this particular receptor in the acidic environment of these vesicles, the antibody is protected against destruction by the degrading enzymes. And then what happens is that these proteins that are not bound to the FcRn receptor are destroyed, immunoglobulins are protected and can be released back into the circulation, and in the physiological higher pH of the blood circulation, the immunoglobulin can be recycled. So therapeutic molecules can be attached to IgG molecules, and you extend the half-life in this way. And a study published last year looked at the factor IX from Biogen IDEC, and this study looked at 4 different treatment groups. And I think if I just go on to the next slide, this is the study from 2013. What it showed was the half-life again is very significantly extended, almost 5-fold. Notice that 90+% of bleeding episodes resolved with, that should say, just 1 injection. And again, no inhibitors or allergic reactions. Now, this factor IX product is already licensed in other parts of the world with the commercial name of Alprolix. It's not licensed in the UK or indeed in Europe at the moment, and I'll come back to that later. Some new data, there are 2 papers actually that are published from the same group in this month's British Journal of Haematology, so September. Again, I can't give you the citation because it's online only at the moment. And I just show you these two papers because what the two papers show is the first one is that this factor 9 linked to immunoglobulins is effective in surgery. 14 major operations, including actually 11 major orthopedic operations, were carried out, and there was really a very good hemostatic response. So that adds new knowledge and experience to bolster our support for this product. And I think also the second paper makes a very important point, which I've quoted here, that switching to this recombinant factor 9 fusion protein for prophylaxis results not only in fewer infusions, which you might have guessed, and lower bleeding rates, but also decreased factor 9 consumption. And I've taken a table in the next slide here from this September publication. What this is showing in the top, and it's perhaps difficult to see it from the back, but this is looking at patients with the various doses, so 50 units per kilogram twice weekly. I'm having difficulty reading it myself here. The number of patients that maintain trough levels of greater than 1%, and that is with the, in green is with conventional factor 9, and in green is with the immunoglobulin version. And if you look at the bottom here, this is people maintaining trough levels greater than 3% with varying dosing regimes. And the point is, with the higher and less frequent dosing, you'll see in green, more people maintain those higher trough levels of 1 or 3%. So it's an important publication. Published also this year was the Biogen IDEC factor 8 molecule. This was the international phase 3 study published from 165 previously transfused patients, including both pediatric and adult patients. Again, patients randomized to 3 groups, either receiving prophylaxis at various 2 doses or on demand. And again, you can see the mean annualized bleed rates in arms 1 and 2 and 3 were respectively. So in other words, when using 25 to 65 units per kilogram every 3 to 5 days, the annualized bleed rate was just 1.6. Twice weekly prophylaxis, 65 units per kilogram, 3.6. And as you might imagine, on demand, the annualized bleed rate was significantly higher. And again, we have clear information that this product is effective in resolving actual bleeds in patients, not just for prophylaxis, but it really gets on top of bleeds as well. This product also has a shorter half-life than the corresponding factor 9 molecule of about just 1.5-fold, actually, compared to the conventional factor 8. There was, interestingly, a clear correlation between the half-life extension and the von Willebrand factor level, which I'll show you in the next slide, and that is of some interest. I think it's very reassuring to note that, again, there were no inhibitors with this product in PTPs, no hypersensitivity reactions, and again, this product is now licensed in quite a few other areas of the world, but no immediate prospect of having this licensed in Europe right now, for reasons I'll come back to. So this correlation between half-life extension and von Willebrand factor level is an interesting one. Let me show you that in graphical format. And if you just look at the bottom for the time being, what this is showing is that this is the von Willebrand antigen level on this axis here, and this is the half-life. What it's showing is that both for conventional recombinant factor 8 product, this was actually Advate, or for the new product, the extension of the half-life is very clearly linked to your baseline von Willebrand factor level. And you will know, I'm sure, that the von Willebrand factor level that you have is linked to your blood group. So people with blood group O tend to have lower levels of von Willebrand factor than people with blood group A, B, and AB. So, whether that might be a selection criterion perhaps in the future, but the people who've got blood groups such as B, A, are going to have higher half-lives than people who have, you would imagine by extrapolation, blood group O. And the final way that you can extend the half-life of this product is the technique that's been exploited by CSL Behring, and they use linking the recombinant molecule to albumin, which they see as the natural alternative to polyethylene glycol. The principle is as follows: to have the recombinant coagulation factor molecule of interest, let's say factor 9, although it's also been done with 7A, and link it to recombinant albumin. And you link it with what's called a linker sequence here, a short linker sequence. Why do you do that? Because you want to keep the albumin away from the coagulation factor protein itself, because that's a very active molecule, and you don't want any physical interference, what's called steric hindrance. So you want something that that protects the coagulation factor molecule from inactivation, but at the same time doesn't interfere with its function. Now, this has an extension of the half-life by the same mechanism that I've talked about earlier, the recycling method. But remember that albumin, why use albumin? Well, it's a natural protein which you have bucket loads of in the body. It's a natural transporter anyway in the body. It's a protein which doesn't really generate immune responses, and it has a long half-life because actually it uses exactly the same recycling mechanism as the immunoglobulin molecule I talked about. And in fact, if you look at that FcRn receptor I talked about when I talked about the Biogen IDEC molecule, here is the FcRn receptor, and on one side there is the binding site for the immunoglobulin, and right on the other side there is an albumin binding site. So the same process is involved in the extension of the half-life of both the Biogen IDEC molecules as the CSL Behring molecules. And here you see the two molecules that CSL Behring have chosen to work with this technique. The first is actually a recombinant factor, activated factor 7, so it's the same product, if you like, as Novo 7, used or aimed at patients with inhibitors. So we've got the 7A molecule linked to albumin with a short linker. And also the technique has been applied to factor 9, which is linked to albumin, and we've got a linker sequence which interestingly incorporates a specific cleavage site which is involved in the activation of factor 9, so that when factor 9 is activated, very conveniently, this linker sequence is eliminated. And Elena Sant'Agostino in Milan has been leading this international trial. The program is called the Prolong 9FP program, and again, so far, some very encouraging results. We see the half-life extension of the factor 9 molecule of about 92 hours, so again a 5-fold extension, and that's just showing it in graphical format. And this has also been done by CSL Behring to try and extend the half-life of recombinant activated factor 7. We know that the product which is most widely used at the moment, the commercially available product, if you like, Novo 7, has a half-life of around 2.5 hours. In a phase 1 study, there is some suggestion that this product, and these are human, obviously volunteers after animal work, the half-life of the recombinant activated factor 7 bound to albumin is significantly extended with a median half-life of around 8.5 hours. Finally, and please bear with me because I know your patience is tested, I just want to talk about CSL's factor 8 molecule. They've chosen a quite different way of extending the half-life of the factor 8 molecule. And just to remind you of the basic structure of the factor 8 molecule, here we have the gene sequence in the various domains, as they're called, the building blocks of the factor 8 molecule, A1, A2, B, A3, and C1 and C2. This is what it looks like in the plasma. After the factor 8 molecule is activated, it is basically divided into two bits, a heavy chain and a light chain. And this is a very unstable molecule which is very rapidly inactivated in the blood in two ways. Firstly, this rather tenuous link, this weak link between the A2 domain here and the A3 domain is broken apart. And then secondly, the heavy chain is broken up. Well, in the factor 8 molecule from CSL Behring, what's happened is that the A2 and the A3 domains are linked much more tightly, and this prevents their dissociation. And what we can say at this stage is preliminary data suggest a half-life extension of the factor 8 molecule by about 1.6-fold, and there's a clinical trial program underway, which is the Affinity program. Now, in the final slide about the actual molecules themselves, I think we've got a clear recurrent theme, and that is that it's possible to extend the half-life of recombinant proteins with 3 different methods: pegylation, Fc fusion, and albumin. With either method, with all 3 methods, you can extend the half-life of factor 9 very readily, 5-fold, but only 2-fold if you're lucky with the factor 8 molecule. And the reason for that is very clearly linked to the carrier role of von Willebrand factor. Factor 9 is free in the blood and circulates freely. Factor 8, on the other hand, is bound to von Willebrand factor. And so even if you have a modified molecule, actually the factor 8 binds to that von Willebrand factor and is carried around by the normal von Willebrand factor. And if you, in experimental animals, knock out both the factor VIII and von Willebrand factor genes, you can extend the half-life of factor VIII five-fold, which shows the importance of the von Willebrand factor molecule. Now let's come on to the more practical issues, which I want to talk about in the second part of my talk. And the first is to hark back to what Mark Skinner, the previous president of WFH, said in his last plenary address, and that is that we really need to rethink about the paradigm model of haemophilia treatment. We've been aiming so far for trough levels of 1%. And I would endorse his words, which I quote here, that this is a trough level which is wholly insufficient. Of course, it's cost that basically limits the treatment. We don't keep people at a normal factor VIII level, unlike diabetes, simply because where we aim for a normal glucose level throughout the week. And I think we need to think about what our trough level should be. And I know Claude Negre is going to talk about this more in his next talk, so I won't tread on his toes. Suffice it to say that I think there is a growing body of medical opinion that levels of 3 to 5% should become the norm. And I think in this context, we need to think at the bottom here about the moderately severe patients, because at the moment we have a paradox where children with severe haemophilia, with prophylaxis, are possible for them to play sports and other things, but children with moderately severe haemophilia are much more restricted because they're not taught prophylaxis, the parents can't often infuse them, and the question is now whether we should actually be treating, I think, patients with moderately severe haemophilia as well. And I think another big issue with the longer-acting products is not only will they be affordable, but should we use the long-acting products for prophylaxis only, or should we use them for prophylaxis and the treatment of breakthrough bleeds? Now, before I talk about that, I just want to remind you that certainly we at the EHC feel very strongly that accessibility of the new products is a priority for patients here in Europe. And we recognise that the regulations regarding licensing restrict or limit the licensing of products here in Europe. That is, you have to submit data on the use of these products in paediatric PTPs, or at least begin, you've got to submit some paediatric PTP data before you get your initial market approval, whereas studies in children are not required by the FDA for licensing there. EHC has already expressed concern about this and predicted this would result in a 2 or 3 year delay, and in fact these concerns have proved justified, as I have already told you. My personal view is that these longer acting products, some of which are already available, as I've said, in the US, are likely to become available here in the EU in the period of 2016 to 2018. That's the time we're talking about. And EHC has lobbied hard on that, and I'd like to give particular credit to Flora Pevandi in Milan, who's done a lot of work on this, and she published a very important paper in Nature earlier this year on this, drawing attention to the problem to a much wider audience. Let's turn to the question of cost, and of course when we do national tendering, it's going to be difficult for us to compare conventional products and the longer acting products in the conventional way of cents or pence per unit. Having said that, the evidence so far suggests that the cost of these products are not going to be prohibitively high, and it's interesting that the pricing of both Alprolix and Eloctate in North America has been based on parity of annual treatment. That isn't weight. In other words, they look at the treatment cost with factor 9 and say, look, our product is going to be pretty much the same. So it isn't an outrageously high price, although bear in mind that prices in the US are generally higher than in many EU countries. And I think this does set an important benchmark for other manufacturers to follow, and given that we've got competition between at least 4 manufacturers of the longer-acting products, I think this will help to keep costs down. Now, turning to the question of whether products should be used, whether we should have the cheaper, if you like, conventional products for breakthrough bleeds, but just use the argument that we use 2 different products. I personally believe that the parity of cost is likely to discourage parallel use of conventional products for top-up treatment of breakthrough bleeds, and indeed patients are going to find it very inconvenient to store and use 2 different products at the same time, and this also introduces the potential for clinical error. But there is also another important point, and that relates to laboratory assay, because all of these new longer-acting products are likely to require the use of their own individual standards for laboratory assays. This is clearly going to disrupt the workload in busy laboratories, And indeed, it may make it difficult to measure the level of factor VIII or factor IX in patients admitted urgently, perhaps for surgery, if they're admitted even to a large hospital at 2 a.m. in the morning. If you don't have the right standards, you won't necessarily get the right result. But bear in mind, it's also not really possible to monitor the level of factor VIII and factor IX reliably in a patient who's using two different types of product. And I think that's also a strong reason not to mix two types of product in the same patient. Now, I know that Claude Negret is going to talk about global assays of hemostasis, and this may provide, once such tests are validated, an answer to this conundrum, but certainly in the immediate future, it isn't going to be possible to reliably monitor the levels of factor VIII and factor IX in a patient receiving two different types of product. And as I reach the conclusion of my talk, I just want to say that I'm impressed in all the various clinical trials I've been involved with in the very strong positive preference patients have experienced for these longer-acting products in clinical trials. And those are some real quotations which I've taken from patients who I asked them to write down their views. And patient opinion and preference is undoubtedly and should be a major force, particularly here in Europe, actually, perhaps less so in other parts of the world, in developing treatment policy. And in that regard, finally, I just want to remind people that we heard the announcement in the Melbourne WFH conference that Biogen IDEC will donate 1 billion units of longer-acting products to WFH to distribute globally. And that's a lot of units, and that's why I spell it out with all those zeros rather than put 10 to the whatever. I mean, that is a lot of zeros. And this is going to be made available in the time period I've set out here. It's interesting that a lot of this product will go to countries currently using very small quantities of factor VIII and factor IX. I hope other companies may feel obliged to follow, but I think a donation on this scale is likely to positively influence demand of the longer-acting products on a global scale, and that people will no longer be satisfied with receiving conventional factor VIII and factor IX, and particularly if the freedom and the quality of life they have is enhanced by having higher trough levels. This is my final slide. As always, I finish within time, and believe me, 38 slides is a lot to get through in that time. Those are my principal conclusions. I'm not going to read them out. I apologise for having put a lot into those slides. I repeat, you're welcome to take copies of the slides. You're welcome to email me and get a PDF copy. Thanks very much.